Hi
Hello!
there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology where I was infecting skin I grew with MRSA. Nowadays, I'm a fact checker for pharmaceutical advertisers and the president of Biotech Without Borders, a nonprofit based in NYC dedicated to the public with the tools of biotech.
My name is Faz, I have a PhD in microbiology, and I've mostly worked on bacteria, but since then I've worked in research integrity, and I'm currently working as an editor for a scientific journal.
Every week, we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen during the week.
And in the next week, we're going to try and put up a poll on at MicroDWAC. So if something has caught your fancy and you want to suggest it to us via Twitter, we'll put it to a poll, and we'll be discussing that towards the
Yeah.
end of the show. So to see what we might cover in a deep dive, where we take one paper and look at it figure by figure to really figure out what it can tell us actually about the questions it asks.
Absolutely. And all of the papers that we discuss on this program, we put in a shared Zotero library linked in the doobly-doo below. And you can browse those folders too to see like stuff that might catch your fancy. And again, if you add us, we'll try to incorporate into a poll or something like that.
And we want to hear from you, so please use the comments or tweet us at MicroDWAC. And boy, do we have a show for you today. First, we might have the answer to why some people appear to be much more resistant to SARS-CoV-2 than others. Not only that, but we are looking at how we're infecting other animals with SARS-CoV-2. And we also look at how funguses can make insects love the cold. And finally, you ever heard about how easy it is to walk and chew gum at the same time? Well, now you can chew gum and fight SARS-CoV-2 at the same time. Want to find out more? Stay tuned.
So the first paper that we have on the docket is we always start the program with some SARS-CoV-2, and we got pre-existing polymerase-specific T cells expand in abortive seronegative SARS-CoV-2.
Yeah, this is a cool. So I think one of the things we've been talking about a bit with our SARS-CoV-2 coverage is what is the ideal immune response to SARS-CoV-2, because that's what we want to like train people with a vaccine. Ideally, want to make. But a lot of the studies that we have, that especially early on, we're looking at people who had like either severe disease or moderate disease. It's been very hard, and
Yeah,
so it's
like I think
quite
early
so.
researchers might have tried to parse the difference, right? Like, could you find some signal inside of our antibodies or our immune cells of a good outcome versus a
Yeah,
bad outcome? Yeah, sorry, that I just wanted to interject there.
yeah. Yeah, and one of the things that I was always wondering is like, well, the people with the best immune response won't ever show positive to SARS-CoV-2. They'd never show up in those so as having exposed to it. So
Yeah, because
the
maybe it was
question
a
is,
very
how do we?
short period in which that they had those markers, but they made so much of
Yeah,
the antibody or the right immune cell that like it was suppressed, and then you don't see that signal.
yeah. So these kind of people, I mean, they may or may not exist. We didn't
Right.
know that until maybe now, because it was very hard to discern them from people who haven't seen SARS-CoV-2 at all. So this paper looks at that. They take so what they do is they take immune serum from healthcare workers who have continuously throughout the outbreak have not been infected at all. They haven't tested positive for any like spike for CR or any specific spike antigens. Um, so this looks at those people, and they take T cells from them to see what kind of epitopes they react to with respect to SARS-CoV-2, and they compare that with pre-pandemic scenario. So serum from people who were there before the pandemic, who could not have possibly been exposed to SARS because SARS didn't exist at that time, and so using that as a control, and they found that there are some people who had a broad response to SARS-CoV-2, but they didn't show up in kind of the traditional testing regimens.
Yeah.
And then they also compared these people to people who had like SARS-CoV-2 infection and looked at the differences in the immune responses between them.
Interesting. So I guess what it's saying is that even though in the course of all their time working, they never got a SARS-CoV-2 positive test, at some point they did encounter the virus, the
Yeah,
antigens. Yep.
yeah. I think the main thing they isolated them is that isolated them that is a lot of these tests that are for SARS-CoV-2 are mainly for the spike. and these people lacked spike memory B cells, so they weren't. So it seemed that they aren't targeting spike, which is probably the big focus for all vaccination research. So they take a look at the epitopes to find out what these people were reacting to, and they found like there's a broad swathe of different. So it can depend a little bit on the HLA type. So that's a type of immune molecule that kind of presents proteins to the immune system. That differs between everybody. But also they found that they had something in common. They were targeting RNA polymerase proteins. So NSP12, NSP7. They're parts of the SARS-CoV-2 genome that work to replicate it, to build RNA. And what they found was that these T cells were much more react to those types of proteins compared to spike.
That's interesting. Strange.
Yeah.
Because, I mean, I think we have seen in some of the epitope mappings that the T cells do respond to a whole bunch of different, it's not just spike, right, that T cells respond to. Because T cells aren't the things that T cells don't make antibodies, right? They're the ones that are detecting, right, some dangerous antigen, but then they are then activating downstream maybe B cells, which do make the antibodies, but maybe also they're becoming cytotoxic, right? And then they're killing off the cells that are showing that they have those little bits. So recognizing those internal proteins, those NSPs, that would be maybe something that's building up for the cytotoxic memory. Because those are things that cells that are infected might be displaying on their surface to say there's this weird protein in me. Is it dangerous? Should I self-destruct?
Yeah.
And a T cell would be responding to that. So that's an interesting finding. Yeah.
Yeah. And obviously the question arises, well, how does this kind of pre-existing immunity occur? And the thing about it is that these RNA polymerases are quite well conserved between different coronaviruses. So the coronavirus RNA polymerase is very similar to seasonal coronaviruses. So what they noticed was that there's a high level of sequence conservation between these coronaviruses. So that means that these people probably were infected with one of those coronaviruses and developed a response that might have been very specific to this RNA polymerase, which means that when coronavirus came along, and RNA polymerase, it comes on very early online during the coronavirus replication process. It's kind of essential which means that that in terms of the timeline, the moment it starts showing that protein is very early on. So if a T cell can recognize it that early, then it can really shut down coronavirus infection much faster than if you're waiting all the way for spike.
That's sort of the hypothesized mechanism of based on seeing the fact that these T cells are recognizing these things, and based on the fact that these people with these T cells never tested positive for never PCR tested positive for SARS-CoV-2 despite all their exposures. There's this idea that maybe they have a different type of their immune response is really good at hitting it at such an early phase because they're somehow cross-reacting with these other coronaviruses that circulate in the past.
Yeah, it's a really interesting study. I mean, they do also could look at another immune correlate. So they looked at alpha-inducible protein 27, which is supposed to be an early innate signature of SARS-CoV-2 to again try, because again, one of the things that's quite hard about this research is trying to prove these people have actually been exposed to SARS-CoV-2, and that their response, because it could be that you're picking up so trying to prove that this isn't just
it actually does something, right?
That they aren't lucky. Yeah.
Just because we found this type of immune response towards these antigens doesn't mean that they ever got exposed and successfully defeated SARS-CoV-2. That event is the hypothesized event in the past.
Yeah. And it's very hard to prove because if they defeated SARS-CoV-2 that thoroughly, then we have to look at other kind of signatures to see whether SARS-CoV-2 was there. So they look at immune correlates that look very similar to other SARS-CoV-2 infections. And of course, they do neutralization assays to see whether their T cells can react to SARS-CoV-2, and they do find that. But this is quite
Yeah,
It's not
that
as big a study as you'd
yeah, that's
like
their final
for
figure,
this
right?
kind of
Figure
thing.
four is like they're like, okay, so we found these T cells. Do they expand if we give them those antigens? And they find that they do, but again, it doesn't. That's not like that. Just shows that they are reacting. They're cross-reactive in this way. But we still don't know like
Yeah.
if those people ever encountered the virus, ever had the infection.
Yeah. I mean, I think that this is an interesting study. I think the way to that would be it'd be quite interesting to see whether people explore vaccines for this and how they go about it, how they go about doing it.
Yeah, I mean, the focus of vaccines that we've had, we've always said like those early stages, the preclinical, or maybe not the preclinical, but the type 1, the phase 1 and phase 2 clinical studies, the endpoints were often, because they weren't doing challenge studies, right? The endpoints were just the correlative immunity, which was antibody titer, neutralizing antibody titer. But this paper would suggest if the hypothesis underlying this paper, right, like is correct, it would suggest that it like I think we talked about this before with T cell, right, when we were saying like what is T cell immunity, right? Like there's another type of immunity that could be productive and helpful in controlling the infection. How do we design vaccines to target that sort of thing? Yeah.
Yeah. I mean I think that the DNA the RNA like vaccines are a very good way to start because that presents the molecules in a way that the T cells are used to. But I think that we do need to do a lot more work to like speak to the T to kind of adjuvant and to like kind of create a signal that the T cells respond to in the way that we like. But this is like there's a lot in the immune system we don't understand yet, so we're still picking this
Yeah.
up as we go
Yeah.
along.
Yeah, because I
But
mean,
I
you
think
brought in
that
that point
yeah,
about like the RNA vaccines, right? But there's not like I don't know if people have a definitive scientific statement that says this is why they work so much, like this is why they're working so much better, right? We know that they are working better in terms of neutralizing antibodies. I'm not sure if we've seen the vaccines stacked up against each other on the T cell axis, but like the why, right, is the question that we'd like to know to inform like the way that we make vaccines in the future.
yeah. And one other point is that if you target this particular protein, you find a way to get T cells to react to it, then you could also potentially have a broad coronavirus vaccine as well, because this protein is conserved amongst lots of different coronavirus species. So that alone might make it a very interesting avenue to explore for more
Yeah,
research.
but so now we're like just adding one on to each of things, but like, but
Go okay.
but with if that NSP though, like this, what they're finding here, it's not the whole protein, right? It's just the epitopes. So like maybe there are sections of that protein that don't react, right? That are unique, but like these are presumably the cross-reactive ones. I don't know if they actually like make a statement on that.
Um,
Right, because
I mean,
just because they're conserved, there's still like
yeah,
much variance between them, right? Like, yeah,
yeah.
the cross reactivity across things like would be like to conserved regions.
Right. Yeah, that is a good point because the proteins themselves are conserved, but if the thing that reacts to is the one part of the protein that is always changing,
Yeah,
then maybe it won't be
yeah.
that amazing.
Proteins are really big, right? I
But
think that's the only point maybe I wanted to just bring to our viewers in case they might have missed that. Like epitopes are really small compared to proteins, which are quite large. Like and when you were talking about like these MHC types, I think we've talked about that before, right? The MHC types introduce a type of bias in the little epitopes that are picked out from the bigger protein whole.
yeah, yeah. So those little like kind of yeah, that's basically it. So this is quite interesting, and I mean, I'm always quite interested in this kind of avenue.
Yeah.
I think I did make us go through a bunch of T cell papers early on in the
No,
outbreak because of
absolutely,
this.
absolutely. No, because and that goes back to the fact that as microbiologists, I think we understand that we learn so much about the human body seeing that interaction between the pathogens on the outside and the inside. And this SARS-CoV-2 pandemic has been this like crazy explosion of information because everyone wants to study SARS-CoV-2, but that also means that we're learning about all of this stuff about human immunity, right? Just in its interaction with this particular virus.
Yeah, Yeah, that's absolutely right. And I think with that, speaking of human immunity,
Hmm.
we've got our next paper, which is a stem loop RNA RIG-I agonist protects against acute and chronic SARS-CoV-2 infection in
Yeah,
mice.
let's go to the immune
Um,
system that we know even better than the human immune system, the mouse immune
yeah,
system.
yeah, we've done so much work over the past century on mouse immunity. It's every, and they're a great model because there are enough similarities there that we can learn something from them.
Absolutely.
Although, uh, yeah, there are some like differences. I mean, one of the differences that's relevant to this paper is that that mice don't get SARS as well as humans.
Right.
They don't have human ACE2, so SARS just doesn't stick to them as well. So they use in this study they use an ACE2 containing
Hmm.
mouse
Hmm.
to study it. Um,
And they're
but I
specifically
think the
looking
thing
at
we
a therapeutic of some sort in this paper.
Yes,
Um,
stem loop RNA RIG-I agonist. So let's break this down. So RIG-I is a pathogen recognizing receptor. So it recognizes double-stranded RNA in the cytosome. So inside of the cell, it sharks around there and it spots some viral RNA. If it spots viral RNA, it sets off a chain reaction to like signal to the immune system like, using interferon that we've got viruses
mhm.
around. Um, so it's designed to clamp onto double-stranded RNA and then, as a result, produce interferon. Um, and I think we've had another number of studies that state that like a big interferon response is really important to protecting against
Hmm.
COVID-19, and also like the viruses do quite a lot to try and stop this response from
Yeah.
happening. So there's a lot of there's a lot of theorized like protein interferences that SARS-CoV-2 does inside the normal cell while it's causing infection, the idea is that it's trying to stop that cell from producing interferon.
Hmm.
Um, so what they do here is they have an interferon like agonist, a RIG-I agonist, so something to make RIG-I angry in the presence
Yeah.
of
So
it.
like SARS-CoV-2 might
Uh,
have the mechanisms to disguise its own RNAs from detection by this system, right? Like it's growing in those membrane vesicles, right? It's
mm,
maybe throwing out some sort of, oh, RIG-I. Yeah, yeah. Wait, um, it's making it's
yeah,
membrane vesicles. Maybe it has like proteins that bind to. I think we saw that it was Nemo or something like that. There is one
yeah.
like there are downstream like signaling pathways from RIG-I that might be being bound to by SARS-CoV-2. But in this therapeutic approach, they're like, well, let's just add our own thing to tickle that particular pathway, and hopefully it will produce the correct immune response.
Yeah. So you got your SARS-CoV-2 going in for like a thief, being like, okay, well like, we've gone past all the alarms, and you send in some idiot to run through and like just trigger
Yeah.
everything. That's basically what this stem loop RNA is doing. It's going off. It's clumsily bumping into all the alarms, so
Hmm.
that the immune system is alerted to it. So that the immune system is forced to produce interferon. And the big question is like, okay, why not just give people interferon?
Hmm.
But that's got its own kind of problems as well because first you need to get the right dosage of it, right?
Hmm.
Um, Also, it's important to get it in the right stage of infection because early on in the infection, interferon is very good at fighting against SARS-CoV-2. At the end of when SARS-CoV-2 is already like triggering the immune system like quite badly, adding more interferon can cause more
Hmm.
damage. So, uh, so I think the idea here is if you let the force the immune system make interferon naturally on its own, then it can regulate it a bit better and produce better
Yeah, and
outcomes.
they do in
So
this paper they do that comparison, right? They do give
yeah,
interferons versus their SL14, and they see better
yeah,
results with their SL14.
yeah, they find really good results with this. So it's a good result when there isn't any weight loss in mice because the first thing that mice do when they get sick is they stop eating and they stop drinking, which makes them lose weight very fast. And that's partly because of their sickness behavior. So like, I mean, same with humans. You lose your appetite
Faz, do
and
you want to
you want to go
scroll
to
down to
bed.
like a figure so people can look? Maybe like
Yeah,
figure
sorry,
four
yeah,
or something like that.
yeah, figure. All right, let's go scroll scroll down to a.
Get a sense of what they're looking at. Yeah, but I mean, these are
That's a
very
good point.
typical mouse infection experiments, right? Testing different interventions. You pick a bunch of time points that you want to look so you can see what your mouse is doing over a certain amount of time. Yeah, you give them the intervention and you see how well they do.
Yeah. In this case, they seem to be doing very well. They've they test this, they benchmark it against other various interferon triggers, and it seems to be doing quite well. This in the way, I guess it's because it's kind of it activates the immune system in the way that SARS-CoV-2 should do it if it wasn't protecting itself. So it's producing that antiviral response. So yeah, this is a way to boost your immune system.
Yeah, well, I mean, it's like this is the preclinical, right? It's kind of like even now, I think maybe this is a good example for folks as well. Is like preclinical work, like it can be quite quick to come together, but then the road from this into actual therapeutics in people, like that can take a long time, and there has to be a lot of money and attention being put on these early stage investigations in order to have any hope of them entering the wider market.
Yeah, actually, I think that's a really important point. And another thing is that with these kind of preclinical models, there is a lot of control over when infection happens and how it happens. And I think, as I mentioned earlier, that interferon is kind of swingy depending on which part of the infection you hit it at. So if you catch it early, it's really good. But if you're a bit too late, then it can actually be either ineffective or bad. And so for preclinical models, it's quite easy to separate those phases out because we control when the mouse gets infected, and we control. But when you're going to a clinical trial, you don't necessarily have that same level of control. So that means that unless it's a really well, you've got everything very like. Sorry, my brain is being destroyed at the moment.
You can see really good results in preclinical, but because like you have so much control in that thing, doesn't mean that those will translate into like when things are more chaotic and in humans.
Yeah, exactly. It doesn't replicate the real situation, because in the real situation, we don't have that level of control. We don't know. There's so much we don't know about the situation. I mean, with these mice, we know exactly how old they are. We know kind of what other mice they've met. I mean, whereas like with humans, we don't know that. And so when you have a treatment that can be this swingy, it doesn't necessarily translate into humans as well. I think we saw this a little bit with remdesivir as well, because remdesivir, very good early on in the infection, might not be that great, and that means that clinical trials are quite difficult to prove that you've caught it in the right window.
Right. Yeah, I was gonna bring exactly that point up. That is the case with remdesivir. And you know, with clinical trials, like that's the whole point of those. Now that we talked about remdesivir, that's the whole point of restricting the eligibility criteria, the inclusion and the exclusion criteria, is an attempt to somehow form up a more controlled population.
Oh.
Is everything fine?
Oh, can you repeat that last thing you said?
With inclusion and exclusion criteria, you're trying to get that control over the human population, so you get the results that you want to see, the positive efficacy results. But also in doing so, you're again artificially imposing this thing, so then when it hits the wider population, right, you may see differing things. And if people remember with the vaccines, there was this big call for making sure that those groups who were included and excluded, that they represented, right, the widest possible swath of our population, especially those who are very vulnerable, the old, the elderly, which are people that might typically be excluded sometimes, right, in populations. And we did see like pregnant women were excluded, right, at the beginning, and it was only later that we sort of picked up the data to say, okay, this is fine, and this is going to work in a wide amount of people. So this is just, I think, all I want to say is there's this trade-off, right, between like the control that you bring over a scenario and how you can like narrow down laser focus to find the result that you want, versus I want to see something that works in a really broad section, right? And now you're exposing yourself to all these different variables that are going to impact, right, whether or not you see the effect.
Yeah, I mean that's why there's a lot of like clinical they find an effect, they find a drug that would work, but it's just not necessarily practical to use in real situations, which is unfortunate because then you have this issue of where you have to deny a drug that would work if you caught it a few weeks earlier. It's a tough situation.
Okay.
But
Let's
speaking of tough situations, as in for
for deer.
deer, I guess.
Speaking of situations, SARS-CoV-2 exposure in wild white-tailed deer. Oh gosh, then there's a Latin name. Odocoileus virginianus.
Yeah, yeah. This is we've talked a little bit about like the exposure of wildlife to SARS-CoV-2. I mean, we've talked about minks. They've had various like preprints come through where people went out and like hunted animals and then found that they had SARS-CoV-2. This one is from the U.S. Department of Agriculture Animal Plant Health Inspection, where they've been now doing a serious surveillance program to see SARS-CoV-2 exposure in white-tailed deer. And what they found, like in 2021, they found that I think about 40% of samples were positive for it. So it seems to be spreading through white-tailed deer populations.
Throw us to figure one, I guess, is like just like all the bulk tests that they have all these tests. They have banked serum that they're able to also go back, right? And see, oh yeah, like none of these deer have seen in the past the SARS-CoV-2 antigens, and then as we go to 2021, it's like, oh yeah, some of these deer have seen things. Their bodies have seen things.
Yeah, I mean, it seems like it is so. So sometimes there's this temptation to think, oh, it's just a spillover. The SARS-CoV-2 might not take traction in other species, but I think here it does seem like it has spread throughout that population.
But so in Figure 2, they lay over their prevalence numbers, right, based on a geographic location, and I mean it does look like the places that there are lots of humans around have the higher prevalence.
Yeah, I mean the one thing that I would like in this kind of study, but obviously they didn't get the chance to have. I think they only managed to get like 6 DNA samples from all of these. But what we'd be ideally having would be kind of some genealogy to see if this is like one SARS-CoV-2 outbreak that stemmed from one crossover event, or that's been like multiple infection events between people and deer.
Yeah, and again, this is sort of the ongoing story and concern, right, of will animals act as a reservoir? So I don't think people have seen the transmission between deer to humans, right? But that would be like the danger factor, and like that's why minks, I think, are put into this sort of danger zone because like from tracking the genealogy, right, like mink to human or human to mink has definitely been seen. I think there have been mink to human.
Mink to human, we definitely saw mink to human because there was like an outbreak around the area where this mink mutations were later seen in other variants as well.
Right.
So yeah,
so this is sort of the surveillance around what animal populations could be reservoirs, right? And really, it should inform policies, right, on how people are interacting with animals and what sort of activities are being allowed in certain areas.
Yeah. Speaking of human interactions with animals,
yes,
we've got our next paper, which is infection with SARS-CoV-2 variant B117 detected in a group of dogs and cats with suspected myocarditis.
Yeah. So this is related, especially in the idea of like animals that have contact with humans are probably more likely, right, to pick up SARS-CoV-2 because if we're the host, the primary host, and it's been spreading across the world, then our contacts with different animals is a point where it can spill over into them. And I found this article linked in Phys, and they were also talking about earlier stuff saying that zoo animals, right, were picking up SARS-CoV-2, right? There was
Yeah.
those tigers and bears or whatever, right, like had SARS-CoV-2. So there's always been, I guess, humans are very concerned about what we have in our own homes and our so-called family, our fur family. Uh, in this paper, they actually are trying to track. They end up tracking this B117 variant in a group of dogs and cats, and so if you go down to really, it's there's not a lot of data here, I would say. So if you go to table 1, you can see like cohort of their entire cohort that they looked at. So it's only a handful of animals, all who've had this they all got diagnosed with myocarditis, which is right a disease of the heart, and we've seen previously right in some cell culture papers that SARS-CoV-2 does infect right this particular tissue. So right, this is a potential link.
Yeah.
This is the potential mechanistic link between seeing this diagnosis and the SARS-CoV-2. And of course, they have some PCR samples from a rectal swab or serology that are why they're drawing attention to this story in the first place, right? Because there's a correlation here between the diagnosis and the presence of SARS-CoV-2 by PCR or by serology. But it's a super small group, right? Like
Yeah.
the correlation is really small. And like they do another sort of correlation later in this paper to try to give us some more weight to the argument too. But again, this is just the correlation of diagnoses of myocarditis, cases of myocarditis in animals. This is figure 1 now. Cases of myocarditis in animals and COVID cases, right? And these types of correlations, you might be able to find tons of them, right, in the world. And so,
I mean,
sorry,
yeah,
you want to say something, Fuzz?
and I was going to say, like, you can definitely see like a crucial difference between the axes that are so on one side you got 12 is the highest number, and the other side you got 140,000.
Yeah. So,
but that doesn't mean that, yeah.
Yeah, so it's hard to say, I think, like, you can show us these types of shapes and say, like, there could be something here. There is that mechanistic nod that we know that it does infect heart tissue, right, of certain, I think the human heart tissue is what we've seen. And then in the rest of the paper is just like, basically like to prove that their diagnoses were myocarditis, right? So they show you like, okay, yeah, these animals did have myocarditis. They're not getting that diagnosis wrong. And then they show you for the few animals they were able to obtain a PCR sample from a rectal swab, they show that is the B117 variant.
Yeah, I mean it's interesting because I feel like we don't often talk about like how vaccination isn't just about protecting your people, but also like your pets. Because I mean myocarditis, I mean yeah. And it's very hard to tell when animals are ill as well, because they tend to like hide it. They tend not to show symptoms until like they're about to go.
Yeah.
It's
I mean,
it's really tough.
That's the way that the article that I found this paper in was trying to encourage people. Like, it's like, okay, maybe you're not getting vaccinated to save the people around you, but you should get vaccinated to make sure your pet doesn't get this. They were trying to push that, but they all were also trying to speak towards this idea that the animals in the zoo are actually getting they're getting a protein-based SARS-CoV-2 vaccine. Some animals, some zoos and some animals are getting a protein-based SARS-CoV-2 vaccine.
Right.
And I think they were trying to get ahead of the idea that maybe people will start asking for this SARS-CoV-2 vaccine for their pets. But that's not going to be possible, is what I read in the article, because of just like they think that in the case, so there's no evidence to say that it's going to jump from your pets back into you, right? Or jump from the zoo animals back into people. I mean, maybe, but again, like this, there's a lot of factors that go into that jump back events, right? And like as we said, we saw it in mink, but have we seen it in deer? Do we see it in zoo animals? That's sort of that's still uncertain. But what zoo animals experience is visitors, right? Like they're seeing like so many humans, right, passing through, and the sort of and then yeah, so that they think that increases the risk, right, to zoo animals, which is why they're pursuing the vaccination for them over pets, where if you got vaccinated, then you're reducing that risk so much for your pet, because how much are you really bringing your pet into contact with so many other people?
Right. Yeah. So yeah, and yeah. So yeah, that brings us to our next study, which is debulking SARS-CoV-2 in saliva using angiotensin converting enzyme 2 in chewing gum to decrease oral virus transmission and infection.
Yeah, so who would have thought anti-SARS-CoV-2 chewing gum is in the pipeline? We've heard about this strategy before, right? Is extracellular ACE2 essentially can act as some sort of decoy, right? That SARS-CoV-2 has the receptor binding domain. It wants to bind ACE2. If you flood that environment with decoy ACE2, then you're going to sop up, right, those particles. And this ACE2 is different. It's a fusion protein. They're fusing it to cholera toxin B, subunit B. And this is something that happens, it actually happens in some chemotherapies, I think. Happens in the research world a lot. Cholera toxin B is not the toxic part of the toxin, but it's the part of the toxin that localizes the toxin to specific cells. So it's very good at binding this particular GM1 receptor. I think that's like ganglioside something or other.
Right.
Yeah. And so like it's present on a lot of cells, epithelial cells. They point out in this paper that it's present on all of these mouth cells, epithelial mouth cells that have ACE2 also have this GM1. So essentially they're localizing all this like decoy ACE2 to the surface of the cells in your mouth. And that is going to prevent SARS-CoV-2, hopefully, from being able to get into those cells, being able to, yeah, and being able to produce productive infectious particles in that environment.
Yeah, and we've heard about ACE2 traps in the context of like nasal sprays and maybe a mouthwash, but I think gum is like a new idea here.
No, wait, you've heard ACE2 in the previous paper. What was that? I thought it was a blood.
I think.
It was a nasal spray.
Oh, I think like I've heard some formulations for nasal spray, but I didn't really, and I didn't think about blood. But I think that makes sense because I think for the mouse studies they would have used blood.
Yeah, they would
because
have.
it's because just for practical reasons. I don't think that it's as easy.
Yeah.
And also the interesting thing about this is like this actually comes from a lab that was studying ACE2. They were trying to grow like lettuce that expressed ACE2 as a treatment for pulmonary disease. I think pulmonary hypertension, and so they engineered some lettuce to express this CTB ACE2 in like the lettuce leaves. And in their previous experiments, they're like feeding it to mice, but now here they like ground it down, extracted those chloroplasts, and then mixed it in a gum formulation.
Yeah, the paper has like the recipe for the gum that they use.
Yeah, we can make it. You can read this paper to learn about how to make gum.
I just pulled up I just wanted to double check because we covered the human ACE decoy paper. I want to see how they delivered it. They did in vitro, so this paper will also do in vitro to show that. Yeah, I mean that of course it works in vitro, right? Like this principle makes sense. But how would they...
Yeah, in vitro it's weird because they had to like add bad powder into it, or so they either ground it up into powder and added that to the in vitro experiments.
Yeah.
Or they put it into a centrifuge to kind of mix everything up.
Yeah. And then I guess in the more applied version, they also made SARS-CoV-2 people chew it and see how much it reduced the amount of...
They didn't actually do that. What they did is they got 15 saliva spoons from COVID-19 patients and they mixed it in with the gum powder.
The gum, right. So it's not...
Although... I should note they do write that the gum tablet chews and performs exactly like conventional chewing gum, based on physical characteristics, but they don't mention how they measured this. So I presume at least one of them must have popped one in their mouth
Right.
and decided to try it.
Well, like you'd
But
wonder, I think what you would want to see is like in chewing, what's the concentration of those like chloroplasts that get like broken up, and like what's that concentration in someone's actual saliva?
yeah, I think they try out like different concentrations of gum to see. Let's see if I can pull down the graphs. Uh, so that's
I'm trying to look at.
so yeah, they got like different concentrations of ACE2 gum. So 50 milligrams. And they found like 50 milligrams is really good at neutralizing.
Yeah. Again, this is the in vitro part.
Yeah, I mean that's yeah, I mean as an additive that's pretty like a lot of like that's a lot of it. Oh, sorry, they're talking about the gum, right? No,
Yeah.
so a minute from there I thought they were like talking about the concentration of ACE2 within the gum.
Oh,
but,
no. It's just the gum, the gum compound. Yeah.
yeah, and they test using like the VSV pseudotype entry. So I think one of the things I really want I haven't really talked about, but like for a lot of these papers they're using pseudotype viruses instead of SARS-CoV-2, which is or like some kind of other thing that it hasn't got live SARS-CoV-2 because I think those techniques are really important in democratizing this process and allowing lots of different science labs that aren't necessarily geared to deal with a SARS-CoV-2 type virus to do experiments like this.
Yeah, like this lab, right? It's a plant lab that was looking for human therapeutics. So they don't have this infectious disease capacity, right, to test the real virus, but they can get like, yeah, the pseudotype allows them to get close.
Yeah.
Yeah.
So yeah. Sorry.
Oh no, I was just saying. So yeah, previously I was just like looking at the ACE2 decoy paper that we read in the past. It was just like they actually only did it. They did it in vitro, or they did it in vivo in mice and in hamsters. And yeah, the dose was, I think, in the blood. So
Yeah.
so this is, oh no, they did intranasal doses.
Right.
Yeah. So it's
So
similar in that same way, right? That if you're able to coat some mucosal surface with like these decoys, then that mucosal surface has some resistance, or should have some resistance. And yeah, the evidence they show, they don't actually, right? They don't make people chew it, so they can't say that it reduced it, but it reduced in their saliva. So if you can liberate the concentrations of gum that they put in there, right, the concentrations of their molecule in the gum in your own mouth, it should replicate this result.
I mean, in theory, because they didn't really have a way to really simulate the chewing action of the gum. And also ACE2 in itself is a drug that has its own effects that I'd be curious about, because this like was initially like this thought of as a treatment for pulmonary hypertension. So I'd be quite interested in seeing whether there are any like other effects of adding this ACE2.
Right. Right. And the other thing I'll say is GM1 is like cholera affects your gut. So cholera toxin subunit B is very effective of localizing things to your gut. And so really it's also going to localize a lot of ACE2 into your gut area. I don't know like what that is all about. Chloroplasts can survive digestion, which is something that happens. So like this also may be delivering some active molecule down there, which again, I'm not really sure what that's going to have an effect on.
And of course, we have to think about like what is as a treatment strategy doing, because it's reducing the amount of like coronavirus in the saliva. Does that mean is it preventing transmission? Is it making the disease less severe? It's quite, I mean, it's not necessarily clear to me how this would fit in a disease control strategy.
Yeah, because if you sneeze, it's not doing anything in your nasopharynx, right? So if you have like a runny nose and you're like sneezing and making aerosols through that pathway, that's only like your mouth is only part of that cavity, right? Like
yeah. So I mean, I like it as an idea. It's a fun idea that I can buy, but I just don't see like, I'm not visualizing how this would like how this could save us from. It's like yet another part of another set of nested strategies against SARS-CoV-2 that I guess could help, but we don't need
Yeah.
to do.
Yeah.
But yeah, I don't know.
Or maybe it's just a platform for dealing with something that is in the mouth, right? Like maybe this is like imagining like cold sore gum or whatever. Like, oh, you gotta like
Yeah.
you have like a weird herpes lesion on your lips and you don't want anyone to get that. And so you're like chewing some gum to try to prevent those things from going. So maybe it's like more of that, like just the idea of it rather than its
Yeah.
application in SARS-CoV-2.
I mean, yeah, this kind of expression platform that they're using for CTBA2 could be used for lots of other proteins as well, theoretically, if they. I mean, so I think that's another really interesting prospect because you can. I mean, maybe I can get much cheaper nicotine gum. Maybe that's what will happen, or other sorts of things you can get in your gum.
Yeah.
CBD gum, maybe. Who knows?
Okay. And that
Okay.
brings us to the end, I think, right, of the papers that we have about SARS-CoV-2.
Yeah.
And the first paper we have in the non-SARS-CoV-2 section is fungal infections lead to shifts in thermal tolerance and voluntary exposure to extreme temperatures in both prey and predator insects.
Is this The Last of Us? Is this?
And so the funny like headline that caught my eye for this one is like just like us, bugs want to get cozy when they're sick. And so what they do is they infect a bunch of bugs with a fungal infection. It's called Boviria bassianea. I'm not familiar with it, but it's a fungal pathogen of insects. They infect these insects with the pathogen, and they see that they don't tolerate excursions to high or low temperatures anymore. They're worse at adapting. So they do both aphids and ladybirds.
Yeah. So ladybirds eat the aphids
Yeah.
and get the fungus that way. And one of the things they talked about in this paper was how a normal insect response to getting a fever would be to find a place of high heat, because they're not exothermic like we are. So they
Yeah.
need to find external sources of heat when they're sick to get that same fever response.
Yes.
But funguses don't like that because they can't survive in high heat. So it seems that when these insects are infected with the fungus, they don't tolerate like those kinds of temperature changes as well, and so
Yep.
they kind of force the insects to live a slightly cooler life.
Yeah, stay in a band
Yeah.
that is more favorable, right, for that virus. And I think the way that they're thinking about this is some sort of metabolic control. They will follow up later and look at like ATP levels, how that modulates that infection. Oh
Sorry,
yeah.
could you repeat that?
The way that they're looking at this is, or the way that they may hypothesize that this effect is achieved is by some sort of metabolism modification, and they'll look at that like by looking at ATP levels.
Right. Yeah.
Yeah,
Yeah, and they find that changes with high fungal
that's right.
loads, and that might be the reason for this change of behavior.
And they have some sort of setup, right, where they set bugs up in like a sort of a home container that has a nice temperature, and then they try to measure how many bugs want to have excursions through like a zone of either elevated temperature or lower temperature. And yeah, if they have a high fungal load, they don't want to do those excursions anymore. I think that's actually number 2. Figure 2 is the excursions figure.
Figure 2. Let's see what was. Um,
Yeah, like it's
yeah.
thermal boldness. So like it's like percentage of the bugs that
Mm,
are staying inside of that are exploring, right, and going across like either cold temperature zones or warm temperature zones.
right. Yeah. So yeah. So the funguses decrease that thermal
Yeah.
boldness. Help them survive over the insects.
Yes. Yes.
Right.
Yeah, so kind
So
of,
I
I
think
think
we're
it's
just
like
gonna.
this is I watched like a real science video about the the zombie fungus or whatever, right? Like that
Mm,
infects ants, and like they were talking about how that fungus like innervates the nerve cells, right, to actually get things to move. I don't know if that's, like, who knows what's happening in this case, but like this is sort of like the larger phenotype that it's capturing in this particular paper, seeing that like infection is influencing behavior. And that is in this case beneficial for the infection. And yeah, just something to think about, I think, like in the world of infectious disease, right? Like there are things that these organisms do to us, right, in order to find a preferred, right, find their preferred habitat, their preferred mode of spread, right, preferred transmission, something like that.
Yeah, I think we're still on for the next paper,
Yes, yeah,
which is
yeah.
yes, uh, coordination of fungal biofilm development by extracellular vesicle
Hmm.
cargo. Um,
So
yeah, so
in
this
a
is
couple of
sorry.
the news articles that we've talked about in the past, like we've talked about extracellular membrane vesicles from bacteria and saying like, oh, the membranes bleb out and that could be a function for antimicrobial resistance. I think we've talked about it in the context of biofilms as well. This is Candida, so this is a fungus, right? Still a single-celled organism, but they also make extracellular vesicles. So they have a different type of cell wall, but they're still able to bleb out these extracellular vesicles. Because they're eukaryotes, they actually have like a whole Golgi apparatus thing going on inside of their cells. It's much more
Mm,
complicated, I'll say, or I'm not sure much more complicated, but they have membrane-bound things inside of their cells that govern these extracellular vesicles. So this escort thing, that's the system of endoplasmic reticulum, Golgi apparatus, vesicles, yeah, the whole process of making these objects inside of the cell.
yeah, I think this is in the context of a biofilm. It seems like using these. So they do this like study with using machine learning to try and categorize knockouts that they made of Candida
Yeah.
to figure out how this extracellular vesicle cargo system is
Yeah,
working.
and I do think they find candidates, right? Where it's like, oh, if we make mutants in these particular genes, it blocks the production of extracellular vesicles. And then they take that and they do some sort of phenotypic analysis and say, oh yeah, like these mutants also don't make as much biofilm.
Yeah, yeah. It seems like these extracellular vesicles are doing like lots of complicated things with the biofilm.
Yeah. And
That
they're
so
full of
like the
proteins, right? Like
yeah.
biofilms, they have to be stabilized, right? Cross all this material that's outside of the cells has to be solidified and cross-linked in some ways. So who knows, right? Like they don't go into detail into any of these particular mutations that they're finding, right? But the idea is that here's a whole bunch of things that like they need to now crack open the mechanism a little bit on how it's stabilizing the biofilm.
Yeah, yeah. Um, yeah, I think that this is there's a lot to pull out here, I think, but it's lots of like little things like
Well,
that
because it
contribute.
is done from as you're saying, it's done from this machine learning perspective where they just picked out all the candidates and then they verified candidates in some phenotypic screen. So I think they do go in later in like the final figure, in figure 4, they like pull out this network diagram of some of the hits that they got, right, where they saw biofilms were impaired and they try to relate them together, right? So I'm not sure what the relations are. I think they might they're the functions, right? So genes, their functions, adhesion, dispersion, and drug susceptibility. But in any case, like this this constellation of genes, right, is improving the production of extracellular vesicles or their cargo, and that has impacts
Mm.
on the way biofilms are being formed.
Yeah, I think the idea is these extracellular vesicles are act as cargo. They information. They convey information that can cause the. So I think there was this idea that you could potentially create your own extracellular vesicles and use that to mess with the network.
Yeah, I guess that's, the discovery of extracellular vesicles, right? Like is big, right? Because you would think that vesicles are supposed to transmit things inside of the cells, right? It's like, oh, that's like moving things from the Golgi to the endoplasmic reticulum. But if you can spit things outside, right, maybe it can merge into another cell and tell that cell to do something. They don't show any of this, right, in terms of like whether or not that's how it's working. It could also just be that the proteins inside of those things are useful for the matrix itself. But yeah.
Yeah, I mean, biofilms are interesting because they're almost the intersection where like singular cellular organisms like almost start making the edge towards being multicellular, like where because they are cooperating, they're coordinating, and they're doing things together.
Yeah, absolutely. Yeah,
Um,
it's a mass of cells, and not all of them are necessarily behaving homogeneously, right? Because like once you cement yourself into a particular structure, now there's relative locations, right, that those cells are in, and that's a really good segue into the next paper, I think.
yeah, yeah, definitely.
Um, yeah, so this one's called Spatial Alanine Metabolism Determines Local Growth Dynamics of E. coli Colonies. Uh,
Yes. Yeah, this.
oh, go ahead if you want to do something.
Oh no, I was going to say that this is again like they go into a lot more detail about the almost the division of labor that is necessary for biofilms to have. Um, in order to survive because the nutrient condition. So you got your so they got this brilliant diagram of the biofilm as it grows, and as it grows, like different parts of the biofilm have different nutrient like
Yeah,
exposures.
because if the,
So
in a it was a colony in this case, right? Not necessarily a biofilm, but like the nutrients are coming from the bottom, right? That's the agar. The agar has all the sugars and stuff that are diffusing upwards, and then the top has all the oxygen, right? Because like that's exposed to air, so oxygen diffuses down. That creates all these different gradients, and I think they do. I think in this one, what they do like they do transcriptomes and also the metabolomes, right? So like the different zones, right, have different are regulating their genes differently, which makes sense because they technically live in different environments, even though it's all just a single colony, right? That's actually a huge space for a single bacterial cell.
yeah, and I think they found something interesting with like an alanine exporter had like had different expression changes depending on like where they were in the colony.
Yes. Yeah. So this is so in cross feeding, right? Cross feeding is this type of division of labor where, right, part of the cells they can make a certain molecule. In this case, they focused on alanine, which is an amino acid, right? Different parts of the cell will make, or different cells in the colony will make alanine and then just shoot it out of the cells. There are other cells they have to import that alanine, right? And then they would absorb and they would be able to use it there. And I think what they are saying in this paper is alanine is made in low oxygen environments. So the cells that are closest to the medium aren't getting as much oxygen, but they're getting tons of nutrients. They're getting sugars, they're getting, right, whatever is in that media. And those cells are the ones that seem to be making alanine, and then the cells in the oxygen environment are the ones that are using it. And they do, yeah, they investigate based on mutants, right? So they have mutants. If they have a mutant that's unable to export alanine anymore, then you get a lot of cell death in the, yeah.
Yeah, and like the shape of the colonies can change as well. Just it's yeah, it gets quite so. I mean, obviously you've got the different environments, and I think they got the different theories of like because I think like alanine, the cells that produce it not just for the reason that it's like good for the whole colony, but I think it's also because they need to get rid of ammonium and other nutrients. So sometimes, so in order to do that, they use excrete alanine almost as a waste product, which is useful for other like organisms in the oxic region that don't have the nutrients, but they do have a need for growth.
Yeah, which is kind of interesting. Like that's so again, like because bacteria have multiple ways of living, right? Like I think we see this in the lab. You can grow bacteria on different substrates, right? Like they don't need a specific thing necessarily, especially like yeah, E. coli has like a pretty wide range of things that it can eat. And yeah, and so within a colony, even though so you said that it's not for the other cells per se, but like it's hard to draw that line, right? Like what is that evolution? Like
Yeah.
the cells themselves are flexible enough that they can exist in two different metabolic regimes, and so they adopt almost complementary regimes based on the resources around them.
Mm. Yeah. Yeah. So I think that brings us to that was our final paper for the
Yeah.
show. Um,
But
yeah, and
we want to choose like four papers to put in the poll, right? For folks to choose from.
yeah. So this will be papers that we looked at over the past month
Mhm.
that we might want to delve into in more
Yeah.
depth.
Was there anything this week that you felt interested in? I like that
Uh,
alanine one.
th- yeah, I think that alanine one is quite an interesting one. Um, Let me s- pull up the Zotero to see what we
Oh,
have.
like some of the things from the past. Uh, Things from the
Yeah, some of these
past.
things.
So
Yeah,
I've tried to remove ones as we talked. There's last week I think we talked about the long-term evolution experiment in yeast.
yeah,
um, I kept a paper about human airways lung on a chips. So like
yeah.
recapitulating the airway, and then they were able to show that flu did mutation in multiple passages on there. So like a model of flu evolution, just all on a cell culture model.
Let's see what else we have here. I mean, I do quite like the pre-existing polymerase-specific T cells, but I do feel like we might have covered everything we were going to say about that.
I
Um,
mean, I think pulling out the it takes some focus to pull out our immunology chops and like refresh them again to explain to people how things work. So yeah, if you want to talk immunology, I think that would be a good choice.
yeah. Let's see what other things there were. Um, because I think like molnupiravir is also something that people are quite interested in. Because
Oh
that's
yeah,
just
the
so.
that's like using I think the title of the YouTube title is like using magnets to figure out how molnupiravir works.
Yeah, I think that could be an interesting way to go about that.
What
Oh yeah, yeah, of course, that magnet paper that was really good.
Right? I think they were like tugging on something. Right? Like, it was like something about how the strand went in, and they were like tugging on it. I might have moved that one
Yeah,
out of this particular document. Do you know? Do you see it in the potentials folder?
I don't. I'm gonna look through my Google Drive notes just to quickly look see
I
where it
think I would
is.
have dropped it in interventions, small molecule, maybe something like that. Date added.
Let's see. Uh,
We can also
so let
search. I can
me.
also search through from molnupiravir. Oh,
Yeah. Okay. Right. So I think that inhibition of SARS-CoV-2 polymerase by nucleotide analogs from single molecule
I see it.
perspective. I think
Cipher et
that's
al.
Yeah, I think so. So it's by eLife.
Yeah, I'll pull that into the potentials.
So it's gonna have good images. Um,
I'm good for that one being like our SARS-CoV-2 choice. Unless you want to choose the
yeah,
immune one as well for our SARS-CoV-2 choice.
I mean we can have more than one SARS-CoV-2
Yep.
choice just to to split the vote.
Yeah, sure. Yeah, two choices on the SARS-CoV-2 side and two choices on the non-SARS-CoV-2 side.
Yeah. Let's see what other things whether
I
caught
mean,
my
this is
eye.
really old, and I know you were excited about this. I'm not sure if we put it in our last vote, but linking plasmid-based beta lactamases to their bacterial hosts using single-cell fusion PCR.
I feel like we. I feel like I'm cool down on that one
Okay,
maybe a little
okay.
bit. Let's see.
Similarly, there's one-cell metabolic phenotyping and sequencing of soil microbiome by Raman-activated gravity-driven encapsulation. It's like putting soil
I
cells
mean,
into bubbles and doing transcriptomes with
yeah,
single ones.
I definitely don't feel like we covered that one in like a huge amount of detail because it's so out of
Yeah,
my wheelhouse. So,
but it's single cell, but
um,
it's single cell bacteria.
yeah, I know, I know. I'm a big fan of single cell. Let me think. How about analysis of SARS-CoV-2 infection dynamic using because there was one on bioluminescence and fluorescence that was also quite interesting for SARS-CoV-2, but that's mostly because of the
Is it
um
correlative multi-scale cryoimaging? Reveals SARS-CoV-2 assembly and
ooh
egress?
no, but that sounds much better as a
Okay.
paper.
Okay. That could
Okay,
be. So we have a
right.
bunch of it. It sounds like we have that. So how do you feel about putting immunology into the mix of things that we're putting for vote?
I think I feel quite. I feel good
Yeah,
about
you
that.
want one to be immunology and one to be molnupiravir,
I,
molnupiravir or correlative
I think.
multi-scale imaging.
Ooh, oh, this is tough. Oh, I think molnupiravir is the one that I'd really want because I think that magnetic study is quite an interesting
Okay.
one to look into. Um, Whereas like with like that kind of cryo-EM, we did cover that in some detail
Yeah.
like a
Yeah, we
while
sort of
back.
looked at the way that it's done at least. We talked about the tech a little bit. Okay, so let's have those. So
Yeah.
those are the two SARS-CoV-2 choices, and then let's have some
Hmm.
non-SARS-CoV-2 choices into the vote. Um,
Yeah.
how did you feel about viral evolution on a airway on a chip?
I feel I feel quite good about that. It's got like lots of it's got evolution on that. It's got microfluidics and
Yep.
airway on a chip. That could be quite
Modeling
interesting.
evolution.
Uh,
I like it's a model
yeah.
of like how viruses are changing, right? Putting that into some system. Okay, and then one more non-SARS-CoV-2, I think.
Let's see.
Oh, I guess we get. Oh, sorry. Go
Okay.
ahead.
So I was going to go with DNA's treatment prevents cerebrospinal fluid block in early experimental pneumococcal meningitis. So that's where
Mm,
neutrophil like extracellular traps block up the brain during meningitis
okay,
infection.
sure. Yeah, let's put that one in.
Yeah.
Yeah, it's like a plumbing thing.
Yeah, that's an interesting thing because yeah, uh, and I think it was one of the first few papers I saw that had like neutrophil extracellular trap like
Hmm.
images of how of them working or not working, depending on how you
Yeah.
define them. Um,
Okay. Well, we'll
but
put
yeah,
that
I think
onto a Twitter poll and send that out, and
yeah,
maybe for a week or 2 weeks we'll try to direct some traffic there. So if people are interested in directing the sort of stuff that we're gonna speak about, that's where you should look. But otherwise,
mm,
we're gonna be off next week for BioSummit,
Bio
yep,
Summit.
which is
Yep.
a conference. But we'll be back the week after that with more cool microbiology news.
Yeah, thank you so much for listening. Um, yeah, we want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. So science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the
I totally
comments.
agree. You can reach out to us over Twitter at microtwjc. We both believe that peer review is a process and that we can all participate in it. So we hope that you've had a good time listening to us ramble on about microbiology today. If you have something to add or found something unclear, let us know in the comments.
It's been a pleasure chatting
Same
with
here,
you, Danny.
Fuzz.
Tune in next week for more microbiology
Well,
content.
a week
Bye.
after the next week.
Oh yeah, week after next week, yeah. But we'll definitely be seeing you then, or you'll be seeing us. One of
See ya.
those.